Greetings everyone. Today we delve into the interesting topic of the biological clock and circadian rhythm in the plant. So let's start. The biological clock regulates numerous plant processes including transpiration and the synthesis of specific enzymes which exhibit oscillations throughout the day. These fluctuations often correspond to changes in light levels, temperature, and relative humidity that occur during the 24-hour cycle of day and night. Interestingly, even under constant conditions within a growth chamber, various physiological processes in plant persistently oscillate with the frequency of approximately 24 hours. For example, the opening and closing of stomata and the production of photosynthetic enzyme continues to display rhythmic behavior. As illustrative example is observed in many legumes where leaves lower in the evening and raise in the morning, that is a movement that persists even when plants are subjected to constant light or darkness. This indicates the inherent nature of the plant's biological clock in orchestrating rhythmic physiological activities regardless of external environmental cues. Now let's discuss about the circadian rhythm. The plants during endogenous rhythm, even when subjected to constant environmental conditions, these rhythm known as circadian rhythm follows a cycle lasting approximately 24 hours. They synchronize with the daily day-night cycle through a process that is called as entrainment. Notably, organisms maintain these rhythms even in extreme environments such as deep mine shafts or satellites, indicating that subtle environmental cues do not trigger them. Contemporary studies propose that the oscillator governing circadian rhythm is internal or endogenous. However, this internal clock aligns with a 24-hour period through synchronization or entrainment facilitated by daily environmental signals. In a constant environment, the circadian rhythm of an organism may exhibit deviations from the typical 24-hour period with free running period spanning from 21 to 27 hours. However, these variations in the free running period do not signify erratic drifting of biological clocks. Rather, they indicate a lack of synchronization with the external environment. When considering biological clock, it's crucial to distinguish between the oscillator or clock and the rhythmic processes it regulates. For example, restraining the movement of leaves on a bean plant temporarily disrupts their rhythmic behavior, but upon release, they swiftly realign to the appropriate position for the given time of day. Despite potential interference with biological rhythm, the underlying clock mechanism continues to function accurately, ticking off time consistently. Now let's discuss some historical background. The Erwin Bunning in 1936 proposed a conceptual framework suggesting that daily rhythm in plants are composed of two distinct phases, that is the photophil phase, also known as the light loving phase, and the scotophil phase referred to as the dark living phase. During the photophil phase, characterized by intense exposure to the light, exhibit heightened photosynthetic activity. This phase is marked by anabolic processes where energy derived from photosynthesis is predominantly utilized for synthesizing organic compounds. In contrast, the scotophil phase, occurring during periods of darkness, is categorized by increased respiratory activity within this phase. Catabolic processes within this phase, catabolic processes become dominant, leading to the breakdown of stored starch into simpler sugars through hydrolytic activity. The scotophil phase is associated with the heightened respiration rates and a shift towards energy release rather than reflecting the plant's adaptation to utilize stored resources for metabolic activities during periods of limited light availability. According to the Bunning's hypothesis, plant rhythm cycle between two phases, that is the photophil and the scotophil, approximately every 12 hours. In condition of constant environmental stability, the photophil phase is presumed to align with the subjective day, while the scotophil phase corresponds to the subjective night. The impact of light on the flowering process is contingent upon the phase at which the light stimulus is encountered. When light is administered during the light sensitive phase of the rhythm, it serves either to induce flowering in long day plants or inhibit the flowering in short day conditions. This phenomena illustrates how the timing of light exposure relative to the plant's internal rhythm influences its developmental processes. For instance, in long day plant, exposure to light during the light sensitive phase facilitate flowering, whereas in short day plants, light exposure during this phase impedes the flowering process. This highlights the intricate interplay between internal circadian rhythm and external environmental cues in regulating plant physiology and development. In an experiment involving Chenopodium rubrum plants, which are categorized as short day plants, needing only a single photoinductive cycle to initiate flowering, a transition to a 72-hour dark period occurred following exposure to photoperiod. During this prolonged dark period, brief 2-minute night breaks were introduced at various intervals before transferring the plants to continuous light conditions. The most significant suppression of flowering was observed when the night break was administered at 33 or 60 hours after the start of the dark period. These time points coincide with that would typically be the scotophil phase or dark living phase in a standard 24-hour light dark cycle. Conversely, when night breaks were applied around 18 and 46 hours after the initiation of the dark period, there was no observed inhibition of the flowering. These time points align with what would typically be the photophil phase or light-loving phase in a 24-hour cycle. This observation indicates an intricate interaction between the process of photoinduction and the endogenous rhythm of the plant, suggesting that the timing of light exposure related to the plant's internal rhythm significantly influences its responsiveness to flowering stimuli. Flowering induction in both long day plants and short day plants is intricately tied to the alignment of light exposure with the appropriate phase of the plant's internal rhythm. Central to this regulatory process is the presence of circadian regulator, a mechanism initially proposed by Irving Bunning. Over time, Bunning's hypothesis has evolved into what is now known as the coincidence model, which provides a more nuanced understanding of the molecular mechanism underlying flowering regulation. According to this model, a critical regulator accumulates within the long day plants, reaching its peak concentration during periods of extending daylight. This regulator crucially requires light for its activation, indicating that its presence coincides with exposure to light. Upon activation, the regulator initiates a cascade of molecular events, ultimately leading to the induction of flowering in long day plants. The coincidence model offers valuable insights into how plants integrate external environmental cues such as light exposure with their internal biological rhythm to precisely time the onset of flowering, thus ensuring reproductive success and adaptation to changing environmental conditions. In Arabidopsis, classified as quantitative long day plant, several genes including Gigantea, constans and flowering locus have been identified as pivotal regulator of the flowering. A breakthrough in understanding flowering control occurred with the discovery of a mutant of Arabidopsis which displayed delayed flowering exclusively under long day conditions while remaining unaffected by the short day conditions. Subsequent investigations led to the isolation of the constans gene, revealing its central role in photoperiodic flowering control. In Arabidopsis, mutant mRNA begins to accumulate, peaking under long day conditions, and is translated upon exposure to light. The stability of the CO protein is augmented by blue and far red light absorbed via cryptochrome and phytochrome A pigments respectively. Co-expression and subsequent FT gene activation occur in companion cells followed by FT protein transport to the shoot apex where it facilitates flowering. Thus, in Arabidopsis, flowering ensues when CO gene transcription and translation coincide with light exposure, specifically under long day conditions. Moreover, in the regulation of flowering in Arabidopsis, there exists a significant overlap known as coincidence between the synthesis of constans mRNA and exposure to daylight. This coincidence allows for the accumulation of active constans protein to levels conducive to the promotion of flowering. Thus, the rhythmicity of constant mRNA accumulation in response to photoperiods coupled with translation into co-protein under independent condition forms the molecular basis for the external coincidence model. Flowering locus T, that is FT, acts as a downstream target gene of a constans and is expressed in companion cell. The regulatory role of constans is mediated through expression of the flowering locus, indicating their independent relationship. The movement of flowering locus from companion cells to sieve elements is facilitated by the endoplasmic reticulum localized protein called the FT interacting protein. Upon arrival in the floral meristem, FT protein translocates into the nucleus where it forms a complex with the BZIP transcription regulator FD and encoded by flowering locus D, that is FD gene. This FT-FD complex then activates the expression of floral meristem identified genes, including MADS box transcription factors such as suppressor of overexpression of constans 1 and AP1. These genes specify that the vegetative shoot meristem of the plant gets differentiated into the floral meristem. Studies focusing on the flowering patterns of rice plants categorized as short day plants have revealed the presence of key regulator genes such as constans and flowering locus, which are conserved between rice and Arabidopsis. However, evolutionary adaptations have resulted in the alteration to the specific regulatory mechanism of these genes in rice, favoring flowering induction under short day conditions. In rice, homologs of CO and FT are represented by the genes that is called as the heading date 1 and heading date 3 respectively. Analogs to FT in Arabidopsis, the expression of HD3A in rice triggers rapid flowering irrespective of the photoperiod. Furthermore, the expression of FT in Arabidopsis and HD3A in rice leads to heightened flowering during inductive photoperiods encompassing both long days and short days respectively. Moreover, in Arabidopsis classified as a long day plant, the synchronization of the constans expression with the right period promotes flowering. However, in rice, a short day plant, the alignment of HD1 expression with the daylight actually suppresses flowering as HD1 functions as suppressor of HD3A. Unlike in Arabidopsis, where the coincidence of CO expression with light facilitates flowering, the absence of alignment between HD1 mRNA expression and daylight in rice prevents the accumulation of HD1 protein. This HD1 protein serves as a repressor of the gene that is responsible for encoding the transmissible floral stimulus, that is HD3A. In rice, consequently, in the absence of HD1 protein repression, HD3A mRNA is expressed and leading to the translocation of the encoded protein to the apical meristem where it initiates flowering. Under long day conditions, which are detected by the phytochrome, the peak of HD1 mRNA expression coincides with daylight, enabling the accumulation of the HD1 repressor protein. As a result, HD3A mRNA remains unexpressed and the plant remains in a vegetative state under long day conditions. This disparity underscores the divergent regulatory mechanism between Arabidopsis and rice in response to photoperiodic cues. Moving forward, the circadian rhythms are characterized by three key parameters that define their cyclic nature. First, the period refers to the time interval between the comparable points in repeating cycle, typically measured as the duration between consecutive peak or trough. These peak and troughs represent the maximum and minimum points of the cycle respectively. Secondly, the phase denotes any point within the cycle identifiable based on its relationship to the rest of the cycle, with the position of peaks and troughs serving as prominent phase points. These serve as recognizable markers within the cyclic phenomena. Lastly, the amplitude of a cycle phenomena is defined as the distance between the peak and trough, representing the magnitude of intensity of the observed oscillation. Notably, while the period remains constant, the amplitude of a biological rhythm can vary, indicating fluctuations in the strength or magnitude of the cyclic phenomena over time. Together, these parameters provide a comprehensive understanding of the characteristics and dynamics of the circadian rhythm. In conditions of the constant light or darkness, circadian rhythm exhibit deviations from a precise 24-hour period, resulting in a drifting pattern related to solar time. This drifting can lead to either a gaining or losing of time depending on whether the period of the rhythm is shorter or longer than 24 hours. However, under natural circumstances, the internal clock or endogenous oscillator is synchronized to a true 24-hour period by environmental cues. These cues, notably the transition from light to dark at dusk and from dark to light at dawn, serve as Zeitgebers, which is German for time givers. Zeitgebers play a pivotal role in entertaining the internal clock to the external day-night cycle. In the absence of such environmental signals, such as when organisms are placed in continuous darkness, the circadian rhythm enters a free running state. In this state, the rhythm reverts to its inherent circadian period, characteristics of the particular organism, emphasizing the dynamic interplay between the internal biological rhythm and external environmental cues. Moreover, the circadian clock functionality hinges on its ability to accurately keep time amidst fluctuating temperatures experienced in natural conditions. Surprisingly, temperature fluctuations have minimal impact, if any, on the period of the free running rhythm, a phenomena referred to as temperature compensation. This remarkable feature allows the circadian clock to maintain constant timekeeping across various temperature ranges. Temperature compensation is achieved through a complex interplay of biochemical processes within the circadian pathway. While all biochemical steps in these pathways are sensitive to temperature, their responses appear to counterbalance each other. For instance, changes in the rates of synthesis of intermediates may be counteracted by parallel changes in the rates of degradation. Consequently, this delicate balance ensures that the steady state levels of clock regulators remain stable despite variations in the temperature. As a result, the circadian clock can effectively regulate biological processes across diverse environmental conditions, highlighting its adaptability and resilience. As you can see in the figure, some characteristics of circadian rhythm. A typical circadian rhythm, the period is the time between comparable points in the repeating cycle. The phase is any point in the repeating cycle recognizable by its relationship with the rest of the cycle. The amplitude is the distance between peak and trough. In the figure B, the suspension of a circadian rhythm in continuous bright light and the release or restarting of the rhythm following transfer to darkness. As you can see in the figure C, a circadian rhythm entertains a 24-hour light dark L to D cycle and its reversion to the free running period 26 hours in this following transfer to continuous darkness. Moreover, the figure D shows typical phase shifting response to light pulse given shortly after transfer to darkness. The rhythm is rephased or delayed without its period being changed. Moving to the next, that is the phase shifting adjust circadian rhythm to different day-night cycle. In circadian rhythm, physiological responses are intricately linked to specific time points of the endogenous oscillator, ensuring that these responses occur at consistent times of the day. Remarkably, a single oscillator can govern multiple circadian rhythms even if they are not synchronized with each other. Despite variations in the duration of light and darkness throughout the season, these physiological responses manage to maintain their timing accuracy. To assess the response of the endogenous oscillator, researchers typically subject to the organism to the continuous darkness and observe its reaction to brief pulses of light, usually lasting less than an hour, administered at different phase points within the free running rhythm. When an organism is synchronized to a 12-hour light and a 12-hour dark cycle and then transition to free running conditions in constant light or darkness, the phase of the rhythm that aligns with the light period of the previous entertaining cycle is termed the subjective day, while the phase corresponding to the dark period is labeled to the subjective night. This intricate system enables organisms to adjust and synchronize their physiological activities with environmental changes, ensuring optimal adaptation and survival. Many circadian rhythms have free running periods that deviate from the 24-hour daily cycle, causing them to become desynchronized from the natural environment. If deprived of environmental cues, humans encounter this desynchronization known as a jet lag when the crossing multiple time zones during air travel. However, over time, our circadian rhythm tend to readjust and become synchronized once again with the external environment. Similarly, plants also possess the capability to reestablish or entrain their circadian synchronization. This process enables both human and plants to adapt to changes in their surroundings and maintain optimal biological functions in alignment with the daily cycle of the light and darkness. Both phytochrome and blue light photoreceptors play essential roles in training the circadian rhythm of the plants. The phytochrome system operates by toggling cellular responses on and off through the PR-PFR switch. In darkness, the phytochrome ratio gradually shifts in favor of the PR form, partly due to the synthesis of a new PR molecules and in certain species through the slow biochemical conversion of PFR to PR. However, when the sun rises, there is a sudden surge in the PFR level triggered by the rapid photoconversion of PR. This daily surge of PFR at dawn serves to reset the biological clock, effectively synchronizing the plant circadian rhythm with the external light dark cycle. This mechanism enables plants to adjust their physiological processes in response to changing environmental conditions, ensuring optimal growth and adaptation. So, in conclusion, we can say that the biological clock and circadian rhythm represent marvels of plant biology, shaping the lives of plants in profound ways. Through a combination of molecular, physiological, and ecological studies, we continue to unravel the mysteries of these intricate timekeeping mechanisms, shedding light on the fundamental principle governing plant life. So, this is all about today's lecture, and this lecture is also the last lecture of the plant physiology and metabolism class. Thank you so much.